Today we're going to be learning about DNA structure. We see these letters down here, and we're going to come back to these later. We're going to come back to those letters later. I want you guys to write this in your book. So this is DNA. It's a double helix. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. That stands for DNA. We see this, like I said, it's like our ladder. We twist around and it goes around like this. So it is our double helix form. It's our double helix form. The first nucleic base is cytosine. So we see cytosine. Like I said, that's not how it's actually shaped, but that's how we document it so we can tell the difference. Now we're going to look at its partner. Its match, its complement is called guanine. So this is the pair. This is the match for cytosine. So the next one is called thymine. It's called thymine. And our last one, the pair with thymine, is adenine. The pair with thymine is adenine. So we have those four pairs together that all match. So here's a picture of DNA. As we see, adenine always goes with thymine. And as such, cytosine always matches with guanine. Is there ever a situation where others can match together? No. So these lines, these helices, are opposite of one another. They are complementary. They are the reciprocal. They are the reciprocal. So that's what we know here, is that these two are opposites together. When we're reading these nucleic bases, we're reading them in groups of 3, just like that video said. So here's our sentence. She has 1 cat. Each grouped in 3. And we can read it pretty well, right? We can read it pretty well. But here's what happens when mutations come about. First, if we have substitution, we might have this sentence happen. Now, can we still read this pretty well? Yeah, we can still read this pretty well. That first word, we're like looking at it like, okay, there's obviously a problem with it, but you might think, oh, it's a typo, it's a spell check. You know, you can come up with it pretty quickly. Regardless, you know something has 1 cat. But here's where it gets dangerous. Let's look at deletion. In this case, I am going to delete that H that's right there. And instead, I'm going to be left with this. Can I read that as easily? No, I have no idea what that sentence is saying. Se, eso, nek, at. I have no idea what the sentence is saying. Same thing with insertion. Here, if I'm going to insert something in, I'm going to put that there. Does that work? Can I read that very well? No. I'm looking at these. I have no idea. If I just presented you guys with those 2 sentences right there, would you have any idea what the original sentence was? No. That's why deletion and insertion have the potential to be the most dangerous of all of them, because it's going to mess up the rest of the DNA down the entire line. It's going to mess up the rest of the DNA down the entire line.